Alright, when we last left off, we did the light-dependent phase.
Mr. Melsick, what happened to your hair?
Alright, so what happens in the light-dependent phase? We got our water coming in along with light energy. They're going to enter the thylakoid where we're going to split our water. And then we're going to the oxygen that comes off of the water is going to wait for another water molecule to split. The two O's are going to get together to make O2, which is a gas that's going to diffuse out of the thylakoid, through the stroma, out of the chloroplast, and then any excess is going to diffuse out of the plant where we get to breathe it. Alright? Yay, yeah. Alright. So then we've got our hydrogen that are left over. These guys are going to be stripped of their electrons. The electrons go into photosystem 2. The protons stay in the thylakoid space. The electrons are going to move down from photosystem 2 to photosystem 1 through the electron transport chain. And while they go through here, protons are going to get transported into the thylakoid space. Which is over there. I might poke you in the eye. Alright. So, we've moved our... No, you don't want to go there. Don't move that stuff. Okay, so now we've got these protons that are building up in here. We're getting a proton gradient inside the thylakoid space. So the protons are going to move out of the ATP synthase. As they go out, they're going to cause it to rotate. As it rotates, it's going to pick up an ADP and a phosphate group, put them together to make ATP. Meanwhile, our electrons got tired. They get to photosystem 1. They get re-energized. You go down a mini electron transport chain to NADP plus reductase. At NADP plus reductase, the electrons are going to meet up with NADP plus a proton and make NADPH. So we're taking our hydrogens from the water and putting them into the NADPH. We've made ATP. And oxygen left us as a waste product. Okay. So now that we're done with the light-dependent reactions, we're going to take the NADPH and we're going to take the ATP and we're going to use them in the Calvin cycle, the next phase of the process. Are you going to close that door? Can you close that door? Okay.
No, I'm not going to.
He's going to close that door. Okay, thank you. All right. So, what's the first thing we need here? Because we've already used light energy and water, we need something else to do photosynthesis. What do we need?
Joe Mama.
Who's Joe?
I don't know. Who's Joe?
Joe Mama.
Joe Biden.
Oh my God! The teacher is enlightened. The teacher is enlightened. Oh my God!
He's going to pick up the line.
The teacher is smart. Oh my God!
So old.
Thank you.
All right. So,
Hey, Mr. Melsick.
what do we need? What do we need? We already used water. What? No one knows it? Carbon dioxide. So, how many carbon dioxides do we need to make one measly glucose?
12.
6.
Oh,
So smart.
All
my
right.
brain.
So up here on this line coming in, I'm going to write 6 CO2. All right, so I've got 6 carbon dioxides coming in, diffusing into my chloroplast.
Yeah, chloroplast.
All right.
Not zoom in.
All right,
So
so
can we
now that my carbon dioxides are here, I'm going to write them again here just so I remember that they come down here. So I've got 6 CO2. All right, these 6 CO2 are going to meet up with that special enzyme. So plus, do you remember what the name of the enzyme was? No. It's the
Coenzyme
most abundant enzyme. Do you remember that?
Mitochondria.
Nope. All right.
Ribosome.
Ribosome. Sounds like Crisco. All right. So
Rubisco.
You're done.
Rubisco, capital R, little u, capital B, little I, little s, capital C, capital O. Rubisco. Alright? Now, Rubisco's going to grab onto the carbon dioxide, and it's also going to grab onto another molecule, which is really our starter molecule of the Calvin cycle. Now, this one I have not told you the name until today. And this one is called RUBP. So what we're going to do is we're going to meet up with 6.
9.
R, capital R, little u, capital B, capital P. So 6 RUBP molecules are going to meet up with the carbon dioxide and the Rubisco. Now, in order to visualize this better, I'm going to draw out the RUBP, but I'm not going to draw the whole molecule because that would be giant and annoying. Alright, so instead I'm going to just make a stylized drawing, which means it's just going to give the impression of RUBP, but not really be RUBP. Right? So, RUBP has 5 carbons. So in order to note that, I'm going to do 1, 2, 3, 4, 5 circles. And it has 2 phosphate groups on it. So at the beginning, I'm going to do a phosphate group. And at the end, I'm going to attach a phosphate group. So I have 6 times 5 is
30.
30
21.
carbons in my 6 RUBPs. All right?
Yeah.
How many carbons am I bringing in?
6.
6. So whatever I end up with, I should have how many carbons?
36.
36. All right.
Do
So
I write that?
it turns out when Rubisco does the transaction here, what it does is it takes a carbon dioxide and it sticks it into the RUBP, making it, for a brief moment, a 6 carbon molecule with 2 phosphates on it. But it turns out that molecule is really unstable. Oh, it also includes these oxygens. So all of the carbon dioxide gets put into an RUBP. And what happens is it's so unstable that within a moment, it snaps in half, right? And so what you get are 12 molecules of what's called PGA, kind of like pro-golf association. Right? Now this PGA, when I draw it, it's got 3 carbons and a phosphate group. Okay. So PGA is a 3-carbon molecule with a phosphate. So my Rubisco has grabbed 6 carbon dioxides, 6 RUBP, and out of that I've made 12 PGA.
Yeah.
Okay. Now, my next step, my next thing I don't want PGA. Alright? I want what we've been talking about, that important molecule called triose phosphate. And now, give me a moment here.
Yeah.
We're going to talk a little bit about triose phosphate. Now, if you
It's
recall,
in trees.
it's extremely important. Every single organic molecule has been triose phosphate at some point in its life, right? Because this is what the plant is going to use to make everything, right? Now, but of course, we scientists have to do stuff that's confusing, all right? So when you are reading your homework, when you are looking at my lectures, when you are going online to try to find help, when they're talking about triose phosphate, they might use this word, phosphoglyceraldehyde, or they might say G3P, or they might say PGAP, or they might say GALP, or they might say glyceraldehyde 3-phosphate.
Yes.
Because all of these things are exactly the same molecule.
Yeah.
Why? Scientists, why?
Because why not? Why not just confuse high school students and college students and any student of this for Pete's sake? Right? Now, why we wouldn't just all get together as scientists and say, let's call it one thing?
No,
It's
you.
so important. It's one name? No. Right. So
Scientists hate
we
you.
have different names for the same thing. Right? Now, for simplicity's sake, because I can't fit triose phosphate,
P-gal.
I'm gonna use G3P,
P-gal.
right?
P-gal
That's
sounds
why
good.
they use it. That's why they have that one other name.
Yeah, because they're lazy, just like me.
Why can't they just call it like?
Alright,
one?
so,
The number one. Or
number
Like-
one, because it's actually not that, so, alright, we'll talk about that in a minute. So I need to get from my PGA to my G3P or triose phosphate. And in order to do that, I've got to go through a series of transactions, right? This is a very simplified drawing of the process, Thomas. What's going on over there? Oh, okay. So there's actually many transactions going on here, but all Mr. Melsick has done is looked at it and said, what goes in, what comes out, that's all I care about. So, in order to manipulate a molecule, I'm going to need energy. So what do you think I'm going to use?
ATP.
ATP. So I'm going to take 12 ATPs, and these guys are going to become 12 ADPs. And then I'm going to also take 12 NADPHs, right? And these guys are going to become 12 NADP plus and 12 phosphate groups. So the ADP, the NADP plus, and the phosphate groups here, these guys are all going to get recycled. They can go back over to the light-dependent reactions to be transmitted, to be transformed back into ATP. Or this guy's gonna go pick up more hydrogen to become an ATPH again. Okay? So, I'm taking my PGA and I'm gonna make G3P. And I'm gonna make 12 of them. 12 G3P. Triose phosphate. And it's got 1, 2, 3 carbons and a phosphate group. Now you might say, well, what's the difference? Right? The difference is the hydrogens. Alright, all those hydrogens that we harvested from breaking the water, they're getting placed into the molecule. Right? PGA doesn't have the hydrogens, but G3P does. Okay?
Wow.
Right. Now, of the 12 G3P that I've painstakingly made here, 2 are excess. So I've got 2 G3P. Right? And these 2 G3P are going to get transported out of the chloroplast. And they're going to go into the cytosol, cytoplasm of the cell, right? Where outside of the chloroplast, they will become C6H12O6,
Oh, 6.
which is glucose. Now, is it only glucose?
No.
No. You remember from that slide I showed you. It's everything. Alright? So we're going to write here just to remember that and /or others. Because we're going to be able to make everything the plant needs from this G3P, from this triose phosphate. Alright? So we can make glucose, we can make all kinds of carbohydrates, we can make the lipids the plant needs, we can make the amino acids for proteins, and we can make nucleic acids, all from G3P. As long as the plant has the HAPSA, has the trace minerals needed to produce these structures, it's fine. Okay, so meanwhile, let's get back up here. So since I lost 2 of my G3P, that means over here I have 10 left over. 10 G3P. Right? These 10 G3P need to be transformed back into 6 RUDP. Right? Now, how many carbons do I have here? 10 times 3. 30. How many carbons do I have up here? 6 times 1, 2, 3, 4, 5.
30.
30. So I'm not adding in any carbons. But how many phosphorus do I have?
10.
10. How many do I have up here?
12.
Wow. So I need to bring in some
2.
phosphorus. And I also need to manipulate the shape and structure and composition of my molecule, right? And quantity. I'm going from 10 to only 6 molecules, right? So what do I need to do all that work? What do I need to use?
ATP.
ATP, all right? So it turns out I'm going to use 6 ATP. And these 6 ATP are going to become 6 ADPs and plus 4 phosphate groups. So those are going to come off. Now, ADP is a recycled thing. These 4 can go back to help make more ATP, but I've lost 2, all right? And these 2 are becoming part of our ADP, and then they are these 2 phosphate groups that are leaving here at the end of the Calvin cycle, right? When the G3P exits. Now, this is why plants have to have phosphorus, right? Plants need phosphorus in the soil to grow. And the reason for this is because in this transaction here, they're replacing the lost phosphorus. And so they're always, for every turn, for every 1 glucose you're making, you're using 2 phosphorus, okay, to do that. So the plant's got to keep it going. So this is all the Calvin cycle. Now, I mentioned before that in here, from going from PPA to G3P, there's a bunch of different transactions going on in here, but it's too much. Who cares? All right?
I do.
And then over here, to get from G3P back to RDPP, there's a bunch of different transactions going on. I don't care.
I do.
But one thing that's missing from here is in the Calvin cycle, and all of those transactions that's going on, what also comes out of the Calvin cycle are 6 H2Os. We make 6 waters. So there are some dehydration and events that go on in here that remove water. Dehydration synthesis events. They're going to take some water out of here. So that's photosynthesis. Do we have questions? Yes? Yeah.
I don't think so.
And there is, if you do all the math, there's still some extra ATP, but the chloroplast uses that to do its daily maintenance kind of stuff. There's just a few extras. All right? Not enough for the plant.
Bless you.
All right? So, that's that.
No.
Now
That's
let's
everything.
start with
Like,
cellular
Exactly.
respiration.
You
What is
not
the
wanna
first
be
step
blessed?
of cellular respiration?
Right?
Flip your
do you
drawings
not want to
over.
be blessed?
What is the first step? Do you remember what the first step of cellular respiration is?
Breathing.
No.
Being
That is
alive.
incidental, not necessary.
Being alive.
Nope. What is it? No, I need glucose because I've made it. What do I need? What is-
Oxygen.
To exist.
Well,
Food.
I need. In the end, I make pyruvate. What is the first step of cellular respiration?
Water. Oxygen.
Stop yelling.
Wow. Don't yell it because you don't know it.
Exactly.
Let me Google this.
If I'm going to use glucose, what do I have to do to it?
Make it.
I already did make it. That was photosynthesis.
What?
I have to what?
Use up the
I
energy.
heard somebody say it.
Eat.
Make it.
No, I heard somebody say break it apart.
Oh, yeah.
Oh.
All right. Break these. Was that you?
Yeah, it's like it.
Oh. Okay. All right. So what do we call breaking sugar? Wow.
Yeah.
This is the first,
Dehydration
this
synthesis.
is the first period that does not know this. Every other period knew this. How is it that you guys did not pay attention?
Dehydration
Sad.
synthesis.
All right. Now, there's a process. It's the first process of cellular respiration. What is it? Look it up. Look at your notes that you didn't bother taking.
Glycolysis.
That's it.
Yeah.
Glycolysis, sugar breaking. So we're going to take the sugar and we're going to break it. Now, where does glycolysis happen?
Mitochondria.
It does not happen in the mitochondria.
Get ready.
Where does glycolysis happen?
Like nerds. In Lego. But what the fuck?
No idea?
No idea.
In the cell.
In the what?
In the cell.
In the cytoplasm or cytosol. Yeah.
Which
There's
is in
a
the
reason-
cell.
There's a reason why this is not inside of the mitochondria.
Oh, big brain.
All right.
Big
So,
brain time.
glycolysis or glycolysis. All right, glycolysis is in the cytosol. All right, not in the mitochondria. All right, so we have our 6-carbon molecule that is the starter for glycolysis, and this is glucose. Right, so C6H12O6, glucose. What's the matter with you?
What?
A
Is that a
lot.
sneeze?
Bless you.
All right, glycolysis happens in the cytosol. I'm going to start with my glucose. Now, if you recall from your lectures that I'm sure you've been diligent reviewing.
Of course.
Diligently reviewing.
Obviously.
Oh, yes, it's obvious because you didn't even know glycolysis.
Yeah, we always do.
We always paid
All right.
attention to the notes.
There's a slide in there that shows you all of the different molecules that you get from glycolysis. It just goes on and on and on, right?
Of course.
But Mr. Milsick said don't waste your time learning that because I don't
Yes.
care. I
Yeah.
only care about what goes in and what comes out, right? And in glycolysis, there's really just 2 phases. There's one phase to get to this molecule, and there's another to get to the final molecule. What was the final product of glycolysis? Do you remember what that was?
Yeah. Cause we.
Alright, well, let's make this intermediate molecule first, shall we? Alright, in order to make this molecule, I'm going to need to do some work. So I need to use some ATP. Alright, so I'm going to take 2 ATP and break my glucose into 2. I'm going to get 2 ADP from it. And I'm going to get these 2 molecules here. Now, what do you think these 2 molecules are? What? They are. Oh my God. It is G3P or triose phosphate. I made 2 G3P, also known as triose phosphate. Okay. So now, in plants, if they really need to, they could just take this G3P and continue down. But we can't do that because we don't eat G3P, we eat glucose. Okay? So our starter molecule is glucose. Now plants can also start with glucose too because they use that as well. Right? So it just depends on what the plant has around. For us though, we're going to start at glucose. And we're going to work our way down to triose phosphate. Then from triose phosphate, we're going to get to our final molecule. Anybody remember what it is yet? Pyruvate, very good.
What
Also known as
pyruvate?
pyruvic acid. So I'm gonna make 2 pyruvates in the end, but in order to do that, I've gotta do some stuff to my triose phosphate. First thing I'm gonna do is I'm going to remove a hydrogen from each molecule. So I'm gonna take 2 NAD pluses, and that's gonna give me 2 NADH molecules. Then the next thing I'm gonna do is make some ATP from it. I'm gonna use those phosphates that are there, but I'm also gonna need some more phosphates. So I'm gonna take 4 ATPs plus 2 phosphate groups, and that's gonna give me 4 ATP. Now I have pyruvate, so I have 2 pyruvate molecules. Alright, so in glycolysis, alright, I'm taking my glucose. I'm going to break it apart using ATP and back into triose phosphate, or G3P. Then I'm going to manipulate that molecule by taking off hydrogen and taking off the phosphates, alright, making a little ATP, and I get pyruvate. So I get NADH, I get a total from the whole process, a total of 2 ATP, because I need to use 2 ATP here, and I make 4. 4 minus 2 is 2. Okay? So, and then my final product is pyruvate. Now this pyruvate, if I have oxygen, is going to be able to be transported into the mitochondria to do aerobic respiration. And if I don't have oxygen, then I'm going to have to figure out a way to recycle my NAD plus molecule to keep it going, and that's called, to keep glycolysis going.